go over them. Number 2, the restriction enzyme BAMHI predictably cuts the following sequence at the locations designated by the stars. Okay. Which of the following DNA helix sequences would be cut into 3 pieces by BAMHI? So you're just checking for the sequence, right? So you want one that you cut into 3 pieces. So this one would be like it's cutting one site, so you would have 2 pieces. This one cut here. And one cut here, and then there's another cut here. So this would be 1, 2, 3, 4 pieces. This one would not actually GGAT. Nope, this one would not be cut. And then this one you gotta cut here. And a cut here. So you would get 1, 2, 3 pieces. So it's D. Does that make sense?
Yeah, I just like saw it wrong.
Okay. Number 7. Just zoom in here. Compact bone contains rings of osteons, each of which contains a central canal housing the blood vessels, which can be accessed only by those osteocytes adjacent to it. Gap junctions, which are tunnels between neighboring cells, then allow the cells that are not adjacent to the central canal to exchange nutrients and waste with the bloodstream. Which statement is true about the exchange of materials with the bloodstream? Hydrophobic carbon dioxide waste must be passed via gap junctions from high concentration to low concentration. So gap junctions are for things that are like that can't pass through the membrane. Since carbon dioxide we know can pass through the membrane, this would not, it doesn't have to be passed via gap junctions. Okay, so it's not A. Hydrophilic Ca2 plus is passed by gap junctions from low to high. So things don't move from low to high unless there is active transport. So this is not correct. Hydrophobic carbon dioxide waste does not require gap junctions to travel from high concentrations to low concentrations. This is true. And then hydrophilic calcium does not require gap junctions. So calcium certainly does require some kind of channel to move through, because it cannot pass the membrane bilayer. So this is not correct. So answer here is C. And then we have 8, 9, 10. The somatic cells in a newly identified sexually reproducing species are found to be octoploidy. Octoploidy means you have 8 sets of homologous chromosomes. And each cell contains 32 chromosomes, slightly fewer than the 46 chromosomes in a somatic human cell. The cell cycle of the species is similar to ours, and the gametes are made during meiosis. 2 gametes will come together at fertilization to create octoploidy offspring. So that should tell you that each gamete should carry 4N. Okay? So how many chromosome segments are present in a somatic cell at the completion of mitosis? Completion of mitosis should be the same. Should be the same here. So completion of mitosis means the next cell cycle has not happened. So we haven't gone through the synthesis phase. Number 9, how many unique non-homologous chromosomes are present in this species? What's your answer? How do you get it? Anyone?
I chose it wrong. I chose A.
What was your rationale for A?
Like 8N equals 32.
Yeah?
So I pick 4.
So that should be right. Did they mark you wrong?
No, I got it right, but like I'm just not really sure if this is correct or not.
Yeah, it's right.
All right.
So 8N means you have 8 pairs of homologous chromosomes, meaning the chromosomes are the same, like they code for the same characteristics. So when it says how many unique non-homologous, basically if you just ask yourself how many groups of 8 are in 32, or divide 32 by 8, or 8N equals 32. And N represents your non-homologous chromosome number, which is 4. Number 10, how many chromosome segments will be in each gamete if you count each homologous member individually? What's the answer here?
16.
Yeah, half of 32. That's the whole point of meiosis, to create gametes that are half in chromosome number. So that when they come together, they make the full chromosome number. Okay, number 12 refers to this. Populations of goldfish in a large isolated pond were studied in 1958 and again in 2008. The fish's pigment level varied from pale white orange fish to dark brown orange fish. The color of each of the fish was recorded in the figure below. Okay. So what kind of evolution is this? What do we call this? Going from this kind of distribution to this. Nobody? What kind of selection is happening? Okay, this is called disruptive selection, okay? Because it disrupts the average value. So number 12, which of the following theories is supported by the evidence? So right away you should know that this pigment varied from pale white orange fish to dark brown. Okay, so everything in between was not really working out for them. So number 12A says the pigment trait in fish demonstrates incomplete dominance. Is this true or not?
I chose A.
The answer is D, though.
Yeah.
Yeah.
Which of? So this statement, which is incomplete dominance, is not necessarily related to this evidence. The pigment trait in fish demonstrates classical dominance. No. Pigment trait in fish demonstrates codominance. No. I mean, I can see why you would think it's this, because it's light versus dark pigment. But this graph doesn't really tell us anything about dominance, okay, or mode of inheritance. So like it tells us that this like medium pigment wasn't really a good trait, and so they diminished, but that's about it. It doesn't tell us the inheritance. So the way we can figure that out is through crosses. Okay, not through this. 15. So it should be D. And then,
Wait, so basically there's not enough evidence to support this, right?
Yeah, you would have to do some crosses.
Okay.
15. Scientists added radioactive samples of calcium and phosphorus to a tree stump of a recently cut tree. They came back to measure the radioactivity after 1 week. The scientists found that 22 different species of plants with 10-meter radius of the stump had radioactive properties. There is no radioactivity detected in the surrounding atmosphere. So nothing in like the air, but everything was in those species. So 15, which of the following questions are the scientists able to answer with this experiment? What elements are provided to plants from soil? That's not what they're testing here. Let's see. Are radioactive isotopes detectable in small amounts? Again, you don't need to measure like plants uptake of radioactive isotopes by plants in order to measure this. So B is out. And then, are radioactive isotopes transported via vascular tissues in plants? Yeah, I mean, this could be something they're testing because they're basically testing the uptake of these substances that they've put in the soil. Or actually, they've put it in the added to the tree stump. So basically, they're testing if it's going to go down or not, right? So yeah, it would test like whether this thing goes down the vascular tissues in plants. And then, do plants return elements to the soil where they are growing rapidly? Where they are growing rapidly. So this tree is not growing rapidly. So that's not something they're testing. So D is out. So answer is C. Does that kind of make sense? You kind of have to do process of elimination with this one. Okay, 18. Which statement best summarizes the role of calcium in skeletal muscle contraction? You guys don't need to know this. So just skip it. 25. It's based on this. So oxygen consumed versus day of germination of peas. If the trend continues, what will be the oxygen consumed on day 7? Looks like a linear, I would say, linear increase. Like 1, 2, 3, 4, 5. On day 7, it should go up by like another 0.1. So every 2 days it's kind of going up by 1. So day 7 should be 0.6. Does that make sense? Who got that wrong?
Me, Tony.
Okay. Do you understand why we choose that?
Yeah.
Okay. Just look at the
I need
trend.
to look at more carefully.
What did you choose?
I chose 0.55.
Oh, I see. Well, close, but not quite. 26, 27, 28. So the unit of contraction within skeletal muscle cells is called the sarcomere. I don't think you need to know any of that. Nope,
So we can skip 26, 27, right?
Yeah, you can definitely skip that one. What is bound to myosin? Yeah. Skip that one. And then 28. Refer to binding. What is this? Calcium chelator. Which of the following graphs shows how it will affect muscle? Okay. I don't think calcium ion required for the binding. Well, yeah, you guys should be able to answer this one. Did anyone get it? Or did you guess it?
I got it wrong. I chose A. Because I have no idea what this question is.
I got D. I think I got it right.
Oh, I got it right.
Okay. And you guys did it by just like reading this question or just guessing?
Yeah.
Yeah.
Okay. Yeah, so one of you explain how you came to the answer here.
Yeah, Harry, can you explain it to me?
Wait. 28 or 26?
28.
Oh, I got that one wrong too.
Wait, I got it right. Let me read. Let me see.
Only one answer here makes sense, guys.
I got it right, but now that I'm reading back the question, I'm low-key getting dizzy.
Okay, so it says over here that calcium ion is required for the binding of myosin to actin. Okay? If a calcium chelator, do you guys know what this is?
No.
Okay, it's a molecule that attaches to another molecule just for the sake of like attaching to it. So,
What's its purpose though? It just attaches to.
I'll give you an example for one purpose that is actually used. So if you have some sort of poisoning of, let's say, a metal that is harmful to your body, because sometimes like maybe in water that you've drank or some substance somehow got into your body and now you need to get rid of that substance. The way we can do that is give you a chelator of that substance so that it binds to the substance so it can't go in your body and do things that are harmful to you. So for example, if you have a metal poisoning, we can add a chelator that'll bind to that metal and your body will get rid of it instead of the metal going into your tissues and starting to do things that are harmful. Okay? So that's one application of a chelator. But the definition is it's a substance that basically binds calcium. Okay? So if a chelator, such as EDTA, binds calcium, then the calcium is not present for the binding of myosin to actin. So you're going to expect no binding of myosin to actin with high EDTA levels. Make sense?
Yeah.
Okay. So then when you look at these graphs, you have nanomolar of EDTA. So this is the concentration of EDTA. And then you have the percent of myosin in different states. So the dark bars are actin myosin attached at their high energy state. The medium color is actin myosin in low energy state, and the lightest color is myosin alone, so not attached to actin. This is what we're going to expect to see a lot when we have high EDTA levels, because myosin cannot bind actin. This graph is like the complete opposite of what we expect. This one, somehow the low, like actin myosin low energy is increasing with increasing EDTA, but you still have actin myosin binding. So this is wrong. This one is just like going up and down. I have no idea what's happening with that one. There's no trend there. And then with the last one, that's where you see what we expect here. Myosin being alone and detached from actin. Does that make sense? So if you were to be given this question on your upcoming test, it's fair play, because even though you may not have learned about muscle tissue and contractions and all that, it doesn't matter. You have enough information to figure this one out. Okay? All right, so let's move forward. This was number 28. Let's look at 29. Okay, acid rain. Which is a true statement about acid rain? It increases the pH in the water and can harm aquatic plants. So does acid increase pH? Yes or no? What?
Nah. No.
No. So this is wrong. High levels of H plus can harm fish hatchlings. This is true. Okay. Acid releases H plus. And then 20, okay, C, it can change the composition of the soil by contributing more hydroxide ions. Do you guys know what that is?
Wait, isn't it just H plus?
No. I'm going to write these down. You guys should take note here. This is called hydronium. Okay? This is called either hydrogen ion or proton or I think that's all they call it. So H plus, you can either call it proton or hydrogen ion. Okay? These two things are essentially the same. You might see this appear or you might see this appear. The reason why they're both the same is because when an acid releases H plus in water, H plus doesn't just float around as H plus, it actually gets attached to another water molecule to give us H3O plus. So in reality, when we say H plus, we're really saying this hydronium ion. But just to make it quicker and easier, we just go with H plus. But we have the understanding that physically it's in the H3O plus form. And then we have hydroxide. Okay? That's OH minus. This is what a base releases. So this is acid. This is the effect of base. Okay? So this is going to be high pH. This is low pH. The lower the pH, the more acidic something is. Okay. It is caused by the release of heavy metals into the atmosphere. No. It's the release of hydronium or hydrogen ions. Okay. Next is 35. Diabetes mellitus is a disease characterized by an inability of the cells to properly produce type 1 or respond, type 2, to insulin. A hormone, so does this make sense so far? Type 1 diabetes is the inability of cells to produce or properly produce insulin, and type 2 diabetes is the inability to respond properly to insulin, which is a hormone produced by the pancreas in response to high levels of blood glucose. Without insulin, glucose accumulates in the blood. In situations of low blood glucose, another pancreatic enzyme, glucagon, is released, which triggers the process of gluconeogenesis, shown on the right side of the pathway below. So this side. The stimulators, activators, or inhibitors of each step are shown with positive or negative signs. Okay. 35. A researcher wants to identify the conditions that would lead to increased production of fructose 1,6-bisphosphate. Which of the following conditions should she make sure to include in the experiment? So let's find fructose 1,6-bisphosphate here. Right there. Actually, so this serves as a stimulator or activator for pyruvate synthesis. But this is really what we need to focus on, not this guy. Okay. So basically we're trying to see how we can increase this. You look at all the arrows that point to this guy and increase the production of this guy. So ATP citrate and H plus do not stimulate fructose 1,6-bisphosphate production. It's only these two. So you're going to look for an answer that includes these two. Not that one. We don't want ATP, not that one. So it's B or C. Citrate inhibits it. So it's not this one. This one contains both of them. AMP and this guy, fructose 2,6-bisphosphate. Okay, so it's C. 36. Patients with type 1 diabetes, so that's the production, they have issues producing insulin, often require insulin injections. So that kind of makes sense because you can't produce insulin, so you just inject insulin so that your body actually has insulin. Which of the following situations would most require an insulin injection? After eating a stalk of celery, so right away when you're reading this, you need to think of a situation that has the highest sugar spike. Because if you have lots of sugar but no insulin in your body, then your tissues cannot uptake glucose. So then you're going to end up with very high levels of glucose in your bloodstream that you can't get rid of, and that's quite dangerous. So you just compare the sugar levels. Okay? So celery, not much sugar in that one. Cookie, lots of sugar. So put a pin in B. And then C, after skipping breakfast. Well, if you're skipping breakfast, you have no sugar in your body, so no. After drinking a lot of, that's actually dangerous. If you put insulin in your body without any sugar in your bloodstream, well, you always have like a basal amount of sugar in your bloodstream. But if you don't have high levels and then suddenly you inject yourself with insulin, then even that low amount of sugar in your bloodstream is going to be removed, and that's also very dangerous for you. So this is the opposite of what we're looking for. After drinking a lot of water. Water, no sugar, so not that one. So it's B. 37. Which of the following situations likely stimulates gluconeogenesis? Gluconeogenesis is the production of glucose. Which of the following situations likely stimulates this? So let's see. If we have high levels of insulin, means you already have a lot of sugar in your bloodstream, a lot of glucose in your bloodstream. So that's not the answer. High levels of F26 bisphosphate, that's the opposite direction of gluconeogenesis. That's glycolysis environment. And then high levels of glucagon. So literally in here, it says another pancreatic enzyme, glucagon, is released, which triggers the process of gluconeogenesis. So this will be your answer. Okay, and then the last one, ADP, you see it pops up over here, and it actually inhibits the process of gluconeogenesis. So that's not right either. Answer is C. Any questions on that one? Okay, moving on. Embryogenesis is a carefully timed and well-organized process as a single-celled zygote divides and grows into hundreds and thousands of cells. A process called differentiation occurs, wherein certain areas of the embryo become specialized to become different types of tissue. As differentiation continues, the level of specificity increases, and the cell potency decreases until highly specialized, unique tissues and organs develop. The figure below shows 12 stages of development of human embryos. So you start with fertilized egg. You got a zygote with like no differentiation, essentially. Cell adhesion, so things start to organize themselves. And then you get a morula, blastocyst, cells start to move around. And then you start to get differentiation. Okay, 38. The inner cell mass is what eventually forms the embryo. Let's see if they've pulled inner cell mass. Yeah, right here. So you see this chunk of cells? That's what forms the embryo. During development, the embryo differentiates into various types of cell layers. Which of the following is not one of them? So you guys, I don't think would know this one. So all of these are actually part of that differentiation. This one is not. So you haven't learned it, so I don't expect you to know that one. But the answer is B.
Wait, do we have to know these or?
No.
Okay. Sounds good.
I think they've removed all of this, right? It wasn't in your textbook.
Yeah.
Okay.
It was never mentioned.
And then 39, a totipotent embryonic cell has the most cell potency. Totipotent means like totally potent, meaning it can become any type of cell. Which of the following is most likely to be totipotent? Right here, when nothing is differentiated. So A. Next is 40. The following diagram demonstrates the ecological succession that occurs in an environment over time as it is colonized by different species. Why does it take 75 years for a beech maple to occur in the figure above? Okay. Beech seeds have a very long period of dormancy prior to germination. Something like that, you wouldn't even have the information to choose this. So that's not an answer. It takes an average of 75 years for conifer trees to become extinct. This, what? So this is kind of describing the opposite of what this diagram is saying. Agriculture was the predominant industry and hardwood trees were removed. Agriculture was a prominent. I mean, again, this is something you wouldn't really have the information to say, but you do see a nice trend over here. So you should look for an answer that kind of matches what you're seeing, not like random things like this. Maple trees grow better in a pine forest than they do in a grassland. This makes more sense because they grow after the pine trees have grown. So it could be because they're just, better suited to that kind of environment than anything before. So D. 41. During labor, pressure on the cervix and oxytocin from a positive feedback loop is shown below. Form a positive feedback loop, sorry. Okay, which part of the following other pathways also demonstrate positive feedback? Okay, so basically you do not need this diagram to answer this question. You just need to know what positive feedback is. So glycolysis leads to the production of ATP. ATP in turn turns off. No, that's not positive feedback. The anterior pituitary releases ACTH. ACTH then causes the adrenal cortex to release glucocorticoids. They then prevent the pituitary from releasing. So prevent, no. That's negative feedback. LH hormone triggers ovulation and the formation of the corpus luteum. So a lot of these things would have been included in your curriculum, but they're removed. So everything you're reading here would have been in the like other curriculum, and you guys would have like understood what this is saying. Right now, you probably have no clue. The corpus luteum secretes progesterone, which inhibits LH. So you see, you can, if you even draw out a little circle or something, and if one thing that's being produced inhibits the original thing, then it's negative feedback, not positive. When a tissue is injured, it releases chemicals that activate platelets. Activated platelets themselves then release chemicals that activate more platelets. This is positive feedback. Okay, so D. A scientist, so 40, no, we don't have 42. It's 43, no, 44. 44, 45, 46. Refer to the following synthetic pathway of N-RNA pyrimidine cytidine 5' triphosphate, CTP. This pathway begins with the condensation of two small molecules by the enzyme aspartate transcarbamylase, ATCase. Okay. So this is the regulation of CTP biosynthesis. Number 44, which of the following is true when the level of CTP is low in a cell? If it's low, since high CTP prevents this, then we have to think, what would low CTP do? So CTP is converted to let me just see, where is CTP, cytidine. CTP is converted to the enzyme. No, this just doesn't make any sense. This is an enzyme. This is the substrate, I think. Where is it? This pathway begins with two small molecules. No, this is not the substrate, but it's... Okay. So basically CTP blocks this enzyme. It is not converted to it. These are two completely different kinds of molecules. B, the metabolic traffic down the pathway increases. Metabolic traffic down the pathway. So what that kind of means is that you have an accumulation of these molecules. So when you have low CTP, this enzyme is not blocked, so you keep producing this stuff. So yeah, B actually makes sense. And then ATCase or A's is inhibited. It's not inhibited with low levels of CTP, so that's wrong. The final product of the pathway is reduced. No, it should be increased, right? So answer here is B. Okay, this enzymatic phenomenon is an example of feedback inhibition. A product of the pathway inhibits something earlier in the pathway. So that's the definition of feedback inhibition. And then 46, how many cytidine 5-triphosphate pyrimidines can be created from 10 phosphates, assuming all other components are present in excess? So you gotta look over here. You have, what does it say? So 10 phosphates. You just got to see how many phosphates you're going to need in this process. So you have 10. You got one phosphate added here. And then another. Okay, you go from monophosphate to triphosphate. So you add 2 more over here. So 3. You need 3 per molecule. I don't see phosphate being added anywhere else. So if you have 10, then you can make a maximum of 3. Right? Because 3 per CTP molecule. You have 10, you can use 9 of those to make 3. Okay, any questions on this one? Okay, moving on, 57. The following graph demonstrates 3 different strategies for survival. We've seen this before. Which of the following is a true statement according to the figure? The extreme negative slope at the beginning for our strategies indicates that they are unlikely to die young. No, it's the opposite. They're likely to die young, but then some of them live longer. B, the constant slope for C strategists indicate that the death rate is constant. Yeah, that's right. So, I mean, some die early, some die very late, and everything in between. And then C, so B is a potential answer. C, the extreme negative slope at the end of the graph for K strategists indicates that they are more likely to die young. No. I mean, this basically says number of individuals surviving in a small lifespan, like the younger years is down here, right? So this is younger years, and a lot of them stay alive. Even over here in their mid years, stay alive. And then suddenly in old age, most of them die. So these are like lions, elephants, things like that. So that's wrong. They don't die young. The slope of 0 for most of the K strategists' lifespan indicates that K strategists have longer lifespans. No, that's not what it means. It just means they don't die early on. Like their lifespan is around 100 years, as you can see. It's like 90 to 100. Because they all die before that. Okay. So would be the constant. Yeah, it would be B. And then 49. If planctomyces is dividing a wait, oh, it's okay. It's referring to this. Was dividing by budding, which of the following will occur? A mitotic spindle will pull chromosome segments to opposite ends of the cell. No, budding is a different process. Enzymes will unwind the helix and copy the entire bacterial genome. Yeah, are these all... Was it a bacterium? Let me find this guy. Okay, it's right here. It is a bacterium. And... yeah, so... it does do that. There's no mitotic spindle in bacteria, so this is wrong. This is right. So bacteria do this in one go because they have relatively short sequences. C, the nuclear envelope will break down. Bacteria don't have nuclear envelope. The mitochondria, they don't have mitochondria. So it's B. And then 50. Which of the following would make the Calvin-Benson cycle unnecessary? You guys don't know what that is. Either Princeton is teaching a whole other level of AP Bio, or the stuff was removed.
I just don't remember going through like those processes as like deep as Princeton has it.
Okay, let's see if the light depend reactions made sugar and ATP, which following will make unnecessary. So light dependent reactions made sugar. Yeah, so the Calvin cycle. So this one does make sense. If plants could make ATP in their electron transport chain. Well, they kind of do. So this is not an answer. They do make ATP. If plants could use ATP to power cellular, they already do. If NADPH could be created by photosystem 1, that doesn't really have anything to do with this. So if the light, yeah, this one would be A. You'll just have to like study the cycle basically. Like review the cycle if you didn't get this one right. 51. In the following cladogram, a common ancestor. Where is the common ancestor? Why does it have a star here? And species derived from it are illustrated. Okay. How many species have 4 or more common ancestors with Iguanodon? Iguanodon is this guy. And it's asking how many species have 4 or more common ancestors with them. 4 or more. So it's going to be common ancestor, this one. this 4 or more. Okay.
Wait, shouldn't it be
Actually,
5?
not this. 1, 2, 3, 4. 1, 2, 3, 4. What the heck? Common ancestor. Where are you getting 5 from? Where are you getting 5 from?
Yeah, the 5. I circled it. Shouldn't it be that?
One, two, 1, 2, 3. How many spaces? 4 or more common ancestors. So, are they saying these are also species? I'm so confused. Okay, 1, 2, 3, 4, 5. Common ancestor. Okay, let's see. One common ancestor with that guy had 2, 4 or more. 2 with that guy. Okay, I think you're reading it wrong. I think I was reading it wrong because so the ones you've circled, like this guy only has 1 common ancestor with this guy. Right? But it's asking for species that have 4 or more common ancestors. Right?
Yeah.
So that one's out. We don't consider this one. This only has 1 common ancestor. This one would have had 2, 3. And then we have 4, 5. I mean, it would be, 2, 3, and then, okay, this one starts 4. 1, 2, 3, 4, 5, 6. Yeah, it would be 6. It should be D. Does that make sense? So this one only has 1 common ancestor, which is this, with this species. So 1 common ancestor. This one has 1, 2 common ancestors. So 2. This one has 3 common ancestors. So all of these are out because we're looking for 4 or more common ancestors. So this one has 4. 1, 2, 3, 4. And this one has 5. This one has 6. This one is 7. This one is... These two have 8. So these ones. So we have 1, 2, 3, 4, 5, 6 species that share 4 or more common ancestors. Got it?
Yeah.
Okay. '52. Which of the following affects higher order protein structure? Ester linkages between amino acids. Amino acids are not joined by ester linkages. Those are fatty acids, so that's wrong. B, amino acid sequence order. Yeah. Yeah, it does affect higher sequence. So you guys have to go through like primary, secondary, tertiary structures and determine what kind of intermolecular or basically what kind of forces are at play there. So let's go through them one by one. For primary, what determines primary structure?
Primary structure?
Yeah, primary structure of proteins.
That's just like the order of amino acids. That determines structure, is it not?
By primary structure, I'm talking about the like 3D form of the protein. What is the primary structure? What's considered the primary structure? It's just a long chain
Yeah.
of amino acids. So what brings those amino acids together in a long chain? What kind of bond? Peptide bonds. You guys need to know this. So primary structure is determined by peptide bonds. Secondary structure. What about that?
H bond.
What?
Hydrogen bond.
Yes. What kind of structures do we have in the secondary?
Like the alpha and beta sheets.
Alpha, beta. So alpha helices, beta pleated sheets. What creates them? Hydrogen bonds. Okay? It's the hydrogen bonds of the amino groups and the carboxylic acid groups in each amino acid. So it has nothing to do with the R groups. Okay? Tertiary structure is where R groups come into play. We start getting like different 3D structures based on the sequence of amino acids. Because why does sequence matter? It's because of the R groups. Okay. Question is, what affects higher order? It's not the hydrogen bonding between R groups. Hydrophobic interactions between side groups on the same polypeptides.
Wait, what is ester linkage again?
Ester
Yeah.
is like this. Like this group is called ester linkage.
Wait, is the O oxygen?
Yeah. Sorry, this is ester. Like that. This is ester. Or sorry, ether. I mean, technically, if this is attached. Well, okay, so here, the thing is, higher order protein structures, we're talking like tertiary, quaternary, but you can think of quaternary because that's the highest order. So what affects quaternary structures is the amino acid sequence, right? Because that determines the R group sequence, which then determines how the 3D shape looks like. So it's the sequence. And then 53, what is the best testable null hypothesis for the above experiment? Null hypothesis. It's going to be, just without even reading the question, I can say it's D. No preference. Who's wondering about this one?
Me.
Okay, what was confusing?
I picked B because like the numbers on that chart like are pretty different. So
that, okay, the null hypothesis does not come from experimental results. It comes before you even do the experiment. Okay? And the null is supposed to say that there is no preference or there is no difference, there is no change as a result of these experimental, variables. The reason why there is a null hypothesis is that we want to compare the results to the null. If the results are different from the null, then we can do like statistical testing and determine whether we can reject the null or not. So there is a reason why null hypotheses, like the base, which is the null, is no preference or no change. And it's to be able to compare the results. All right. Next is 55. Okay. If the mRNA sequence transcribed from one of the above sequences is this, what is the sequence of the coding strand of DNA? It should be exactly the same. If the mRNA above.
Wait, shouldn't it be 3' to 5' because like anti-parallel?
2. Okay, so these two are DNA sequences. And this is an mRNA sequence that came from one of these, right? from one of the above sequences. Okay. And the question is, what's the sequence of the coding strand? The template strand has to be antiparallel. The coding strand is exactly the same as this. Well, of course, without the uracils. So coding strand is always exactly the same as the mRNA. Even the directionality is the same. So it should be this one. C. So it would be this one. So when the RNA polymerase comes in, it's going to bind this strand. Okay? And it's going to go in the opposite direction and produce this. 57. Blood pressure is determined by the volume of the blood and the peripheral resistance of the blood vessels. This sounds like stuff you don't need to know, but let's see. Blood volume is dependent upon hydration level and osmotic pressure within the blood. Peripheral resistance refers to the volume of blood vessels, which is dependent upon constriction and dilation of arteries as blood flow is maximized and minimized in response to the body's needs. Which of the following would both help to raise blood pressure? Constriction of blood vessels and decreasing salt intake. So let me see, blood pressure determined by the volume of blood and the peripheral resistance. So the blood vessels, basically, if you constrict the blood vessels, you're going to increase the pressure because now the blood has less volume, so there's more pressure on it. So it's one of these, A or C, not dilation is wrong. And then you're going to look at the salt intake. So blood volume is dependent upon hydration level and osmotic pressure within the blood. So how do we increase osmotic pressure? So we need to increase the salt inside the blood vessels, because if we have an increased solute concentration, then that means we're going to pull water into the blood vessels. So it should be C. Does that make sense?
Yeah.
Okay. 59. If it rained 120 inches, what would you project the primary? Okay. That's based on this one. The rainfall and biomass of several trophic levels in an ecosystem were measured over several years. The results are shown in the graph below. Okay. If it rained 120 inches. Let's see, 120 up here. What would you project the primary consumer biomass to be? Rainfall biomass, producers, primary rainfall. You project the primary consumer biomass to be. So clearly, so we have the rainfall graph right here. Clearly, there is some dependence between all these. So the producers, primary consumers, and all that. You already know primary consumers depend on producers as a source of food. So they're going to depend on each other. But the question is, does rainfall affect producers, which then affect consumers? The answer would be yes, because as rainfall increases, we see an increase in producer population, and as rainfall decreases, we see exactly a drop of producers as well. Literally the same as the rainfall amounts. So what this is saying is that if we increase rainfall a bit higher than this, what do you think would happen to the primary consumer biomass? So primary consumers are down here. Okay, and the tip that coincides with this tip of the rainfall, down here, is right here. So basically we're saying if we were to just bump this up to 120 for rainfall, where would this tip go? So primary consumers would increase above, the biomass would increase above 150. Okay, so it's none of these answers. It should be A. Does that make sense?
Yeah.
Okay, and last question. Which of the following graphs best depicts the projected biomass of secondary consumers if they were measured? Okay, primary, secondary, they should all correlate with each other. And you know that biomass decreases as you go up trophic levels. So the biggest biomass is going to be producers, which is this dark line. And then below that should be primary consumers, this line. And then below that should be secondary consumers, this line. So A looks right to me. And then this one, so secondary consumers cannot be greater in biomass than primary consumers. This is wrong. Over here, you see secondary consumers even greater in biomass than producers. Wrong. This, you see secondary and primary consumers at the same levels. Again, that's wrong. So it's going to be A. Any questions?